Yeah, thank you very much for very kind introductions. It's really a great honor to be here talking to this distinguished audience. I particularly appreciate the opportunity to present our work here, as well as to be able to hear all the exciting work carried out here in Taiwan and reconnect with the Taiwan's research effort. As the Vice President described, that my work carried out primarily using these guys, this little guy, Drosophila, as a model organism. For the last couple of decades, we've been focused on trying to figure out what the brain structure, how does the brain work, and really how is the brain constructed during development. So today I would like to share with you primarily on the development aspect of that, focus on this particular neuron. The name is not very important, it's called DM8, but significance will come a bit later. So let me start with a very brief introduction for people who are not in neuroscience. So all of us know that brain is really complicated, but it's really hard to appreciate how complicated this is. Okay, so shown here is the EM reconstructions of a really small piece of the brain piece. This is like about 1% of the 1 milli-cube structures that cut out from the fly. It's probably the most comprehensive connectivity, connectons map that we can obtain in any kind of organism. But you can probably already appreciate all these complex wirings. This is about 300, some 300 neurons making a huge number of synapses. So we know that this complex network, neural network, is capable of receiving information from environments and learns and process information and eventually to guide animal behavior. So we also know that during development, the information from our genome, which arguably has much less information compared with the complexity of the brains, that use the information in the genome to instruct the construction of the neural circuit. So this is really sort of a big conceptually question is how can the information, with much less information contained in our genomes, can really instruct the complicated structures such as our brain. Okay, so today I would like to focus on our more recent effort for the last about 18 years, try to figure out how, from the perspective of the postsynaptic, how dendrite, which is the receiving end of the neural circuit, how dendrite patterns, and how the neural circuit motif forms, and how the synaptic specificity is assigned using the Drosophila as a model organism. So I believe it's Louis Solomon, is a famous architect, American architects first coined this phrase, form ever follow functions. What really means is that, well, what American would say is that, it's pretty is what pretty does, really. Okay, so in neuroscience, probably one of the most famous structures is these Purkinje cells. That has a really beautiful fan-shaped structure of that. The work done in Cholowski's lab actually done quite beautiful computational work to demonstrate that the reason why the Purkinje cells forming such a beautiful fan-shaped structure with certain kind of complexity within the dendritic arbor here is really try to receive as many different type of efforts coming out from the parallel fiber here. So this is probably the only one really good example to tell you about where we can actually, from the structure, and learn much more about the functions, but we can also see in a more sort of superficial way in a particular system when we work on the fly visual system, where you have this individual type of the visual neurons that process different visual information. But you can also appreciate that each different type of this neuron has a specific morphology, which we can recognize by our eyes. Okay, and this region of the brains in flies called medulla is more equivalent to our retina, particularly inner plexiform layers in retina of ours. And you can see that dendrite elaborate into different layers in the brain because they are also layering structures such as our cortex and the retina. And the reason why these all come to a different layer because they want to receive the information coming out from the photoreceptors, and first of all the interneuron coming in here, the color here in red. Okay, so this is another type of the form-follow function. The dendrite need to actually try to match with the presynaptic terminals. Okay, so in another level of conceptual idea is that you can imagine where the dendrite go. That you will determine what type of information it's going to receive, and determine how the information signal can be integrated in the nervous system. Okay, and also the size of the dendritic field that will determine how many of such recipients that can actually take information. Okay. So from a sort of a biomedical perspective, that studying the dendritic morphologies and the synaptic specificity has important implications. And here I list a bunch of pyramidal neurons that have been traced out from a different kind of neuropsychiatric diseases that you might recognize. They all are genetic based, and many of them, most of them actually has a developmental origins. And the list actually go on much longer than that. Things such as schizophrenia, we can also recognize now, is actually statistically different in terms of their dendritic morphology. So taking, for example, the Angelman syndrome level, this is actually a trace of the pyramidal neuron from the patients. You see the dendrites arborized in this very different way. And for example, the Rett syndrome, here that you see a fewer of dendritic arbors and less of the dendritic spines, so you can see here. And one of the reasons why I choose this small number of these diseases are because of that, or being implicated the TOR pathway, the target of rapamycin pathway had been implicated in the disruption of this series of diseases, which I will come back to at the very end of my talk. Yeah. So the goal of that, the last 10 years really want to set up this particular system and then use it to understand what's the molecular mechanism that determine the dendritic patterning, and then how the dendritic patterning defects, such as we find in the disease situation, can actually lead to connectivity defect and eventually to a functional deficit, which is really what we look at it from a clinical perspective. Okay. So let me show you some real data from the lab now. So the particular neuron that we want to describe briefly today, these are the so-called projection neurons, the TM1 to 920s. And as you see, this is the confocal image of that. The cell body is somewhere here being cut out, and the axon coming down and then trying to elaborate in green color, and then the axon continue to go and send information to a higher visual center here, and dendrite. You can see the different type of the neuron arborized dendrite in different fashions. And what you can appreciate is that this region, this group of the neuron are involved in a motion detection pathway, which we and others has worked on for quite some time. They match the dendritic arbor to actually receive the information from the laminar neuron here called L2 and L4. Again, the name's not really important. And for example, this part of the neuron are involved in more on the color vision here on TM20, which we'll spend some time on the elaborate dendrite primarily in this particular layer called L3, M3, and receive input from the L3 neurons. And this is the neuron, the DMA neuron, one of the amicron neuron. They involve in detecting the very deep UV light, which we have identified from the behavioral paradigms here. Okay. So one can then first ask the question about dendrite. We can look at its shape and say, well, this looks different from that. It's really very hard to actually differentiate the different shape of that. So we want to computerize that, try to be systematic about that. So we can register, scatterize the structure and register that into what you see here, the different type of the projection neuron. And then we can then identify what is it really different from, what's the main feature that allow us to differentiate them from one another. So for example, we can see that they all start their dendrite elaboration from roughly, all roughly in this M3 layers. And we can then see that they actually project their dendrite in a different direction. For example, the TM20 neuron, they project dendrite from the anterior end toward the posterior side, whether the other three guys project the opposite direction, as you can see from this polarized plot here. So they have a planar projection difference. And finally, it's the last bit that I already described, the more quantification of that they elaborate to different layers and to allow them to receive different informations from different efferents. Okay, and these neurons all restrict their dendritic arbor into a single column where they actually process a single pixel of information. Okay. So it's probably easier to see from the opposite direction. These are individual photoreceptors that represent each individual visual pixel, and the dendrite elaborate primarily conform to one single column, while the other neuron, for example, the amicron neuron, elaborate a large dendritic arbor to cover about 14 7 photoreceptors. Okay, so they have a size difference. Yeah. So from the morphological analysis, quite a few years ago, we made this hypothesis, is that it's probably take at least 4 steps to read the environment and to form the proper shape of the dendrites. And probably they start with initiating the membrane elaboration from a specific layer and to project in the anterior or posterior direction and or toward a preferred layer, and eventually to elaborate the shape of the dendrite with appropriate size as well as in appropriate complexity here. So in Drosophila, one of the advantages is that we can carry out mosaic analysis. We can remove gene function only in a specific type of neuron, namely, for example, in this case, the TM20 neuron, while the rest of the environment is wild type. And this allows us to really identify gene that function cell autonomously within the TM20 neuron. And with that, we can identify a series of the genes. This is a kind of old list, but this actually go on as we continue screening, finish up the screening. But what we can learn is that it seems to be a specific new molecule that involved in a specific process of that. When you disrupt earlier process, the tendency is to disrupt later processing as well. And I'm going to tell you briefly about the anti-hearing involved in the early part, and then later we'll focus on the size change in terms of dendrite. So removing the anti-hearings from the TM20 neuron alone will actually shift the main dendritic branching point from the M3 layer into M2 layers. So we know that anti-hearing is an evolutionarily conserved molecule. It's a classical coherence, so it contains this cytoplasmic region bind to a catenanes, alpha and beta catenanes, and signal to actin cytoskeletons. We have shown that if you remove this part, then the molecule no longer function in here. So it's not just a adhesion part that carry out by the extracellular domain, but also the signaling part of that. And removing that not only shift the main branching point, also shift the main terminal point as shown here, and then also have some disruption at the planar projection directions. Yeah. Okay. So to cut a long story short, that if this is the interactions, homophilic interaction carried out by the extracellular domain, we got to have the partner. And we can then, we've identified the partners turned out to be a laminar neuron L3, so we can remove the coherent from L3 and also recapitulate the phenotypes of the TM20. So we are now working under the hypothesis that to initiate dendrite to extend in a specific layer, that you need an adhesion that communicate from their target, their presynaptic partners, and in this case is L3, forming a homophilic interactions with the TM20. Okay, and that requires a signaling process to regulate cytoskeleton, where they extend through this point and to further down. And later on, the anti-hearing also mediate the synaptic stabilization, which I don't have time to talk about. But during the development, the TM20 dendrite will further receive a signal from another presynaptic partner that secrete a morphogen called activin. Okay, and this is actually carry out a very different function from the anticoagulants. So removing the activin from the RA photoreceptors, okay, or removing the activin receptor BABU or downstream transcription factor SMAD2 all have very similar phenotypes. Namely, that the TM20 neuron now extend further, they become bigger, okay, extend invading neighboring colonies here. We can then calculate the statistic and shows that the number of branches reduced and the average segment length increase. So one can, there could be this phenomena could be due to the reduction of the dendritic branches frequency or reducing the dendritic terminations. So we can then use the Kaplan-Meier's statistic to actually differentiate these two possibilities. Okay, so this is really what most of you familiar with. This is actually the survival curve. It's just looking at, it's basically looking at the branching and the termination of two censored process. So if you remember from medical school training, then really is that the slope of this determine the frequency. For example, is the dendritic termination or branching. As you can see that the branching frequency didn't really change very much, but termination frequency reduced significantly. Okay. So when you have a larger dendritic field, you actually have more synapse formation. To test that, we use a method that was originally developed by Corey Thomas' group. It's called GRASP. Many of you might have heard about it. The idea is fairly simple: to cut a GFP green fluorescence protein into two halves, and one each expressed in the presumptive partners, one in presynaptic partner, the other in postsynaptic partner. In this case, one in RA, the other is in TM20 neurons. And then when these two cells contact with one another, the GFP will be reconstituted and you get back the fluorescence. So you know that these two cells contact. Okay, so using this method, we can show that in wild type, the context is all constrained within one single column. That means that this TM20, wild type TM20, still only looking at a single pixel in the outside world. While if you remove the receptor BABU, the dendrite becomes bigger. Now it's not making contacts and the actually making synapses with their neighbors. So the end result is that you got a bigger dendrite, you lost the one-to-one correspondence. You now become more receptive to actually to the neighbor's information. The signal conceivably become blurred. So the activins not only expressed in RA, but photoreceptor, but also expressed in the other photoreceptor R7 is the UV sensing photoreceptors. And R7 is from the in situ hybridization. R7 secreted activins, and several years ago we showed that this autocrine, through the autocrine mechanism, control the exon guidance and prevent the exon from invading into the neighbors. We further shows that this activin does two jobs. It also provides us a paracrine information, and binds onto the BABU receptor onto its postsynaptic partner DM8 neurons. And the function is very similar from what we have identified in TM20 neuron. One can remove the R7 to remove the activin source, or remove the receptor BABU, or even overexpressing activins, and also result in a very consistent idea that getting rid of activin signaling, then you make the dendrite become bigger. So this is an inhibitory signal, and when you overexpress activins, then you make the dendrite smaller. So it's instructive information of that. Okay, and this is some statistic to show you the result. And I should mention that the activin secreted from R7 cannot be replaced, interchanged with the activin secreted by RA photoreceptor, even though their axon terminals are only a couple of microns away from one another. So this morphogen functions in the brain really in a very short distance. Okay. So last bit of the information that to tell you about is the insulin Tor signaling, which I promised at the beginning. So the other signaling pathway that regulating the size of this dendritic aberration turned out to be the Tor pathway and as through insulin receptors. Okay, so many of you might have already know that similar to vertebrates in Drosophila, also the insulin receptor, once it binds to the insulin like growth factors or insulin, that activate phosphorylated and induce the membrane association, receptor association of the delta protein Chico on the DAC, and this actually goes through a pathway for axonal guidance to through regulation of actin cytoskeleton, and the Chico goes through the classical pathway through the PI3 kinase all the way to the Tor, the target of rapamycin, and it does many different things here, including transcription, translation, and control here. So what we have shown is that if you remove the positive regulator of this pathway, shown here in purple, and you reduce the size of the dendritic arbor in the TM20 neurons. If you remove the negative regulator in this pathway, and the dendrite become larger. So this is the positive regulation of the dendritic arbors. And we also further identify out of all possible multiple candidates of the target of the Tor, turned out to be the important one is one transcription factor called SREBP. So some of you might know that Tor in general described into a Tor1 and Tor2 pathway, and it does regulate very different type of aspect of that. And the particular Tor1 involved in protein synthesis through the S6KF1BP in control the translation, and also several transcription factors, including the SREBP, a steroid hormone, a steroid regulation element binding proteins, and also regulate autophages. So what I find that if you remove other of the pathway, it doesn't really change the size of the dendritic arbor. But if you remove SREBP, you phenocopy the Tor signaling pathway, suggesting that the SREBP is downstream of the Tor, and more importantly, we can actually rescue the Tor phenotype by overexpressing SREBP, and this is a genetic epistasis. Yeah. So similar to what I showed you with the TM20, we also like to know what if the change of the dendritic arbor size do actually change synaptic connections, and perhaps more importantly, do they actually change the synapse function? So we have recently developed another technique that modified the original glass techniques that built by Corey Bauman's now to be able to actually test active synapses. So in this particular modification, that we now hook up the half of the GFP to a synaptic bramines. So normally they only precise constraint in the synaptic vesicle, only during neural firings, and they're exposed onto the extracellular membranes, and then allow them to be able to interact with the other partner. And the other half of them we also modify, and instead of just the membrane protein, simple transmembrane protein, that we actually hook it up to a neurotransmitter receptor, and in this case it's histamine-gated chloride channels. So they allow us to determine whether the synapse is really onto a specific, using the neurospecific neurotransmitters. So on the left side is a contact simple glass method that's shown here. You can see that this DMA neuron color in red and make contact with about 14 of the R7 photoreceptor as shown with this reconstitute GFP fluorescence activities here. But when you look at the synapse activity, you'll see that most of the activity they concentrate in the central correlates here, while the rest of that I showed us only a small spot here. So this is consistent with our EM reconstruction that I'm showing that the majority of the synapses are actually constrained in the central part of the columns. Okay, I also tell you that this actually represents pretty well about what the active synapse distribution look like. So using this method, we can then show that indeed when you have a larger dendrite, by removing the negative regulator of the TOR pathway, then you get larger dendrite, you get more context. But surprisingly, you get a very different synaptic distributions. And now you get more context, but a larger dot in the neighbors. And perhaps more interesting about is the TOR pathway. When you remove the TOR, you get smaller dendrite. You get about the right number of the context from the simple glass method. That activity that you see essentially zero. So what it does that to tell you is that the TOR pathway likely to have a dual function, not just changing the size of the dendrite, but also affect the synapse, the active synapse formation. And I think that's probably something that we need to look into, start looking into all the diseases that involve in the TOR, dysregulation of the TOR. And it is shown here with the statistic. Okay. So the last bit I want to tell you about is that where is the insulin receptor located? Okay, and how is the signaling part? So, so one of the ambition in development is really want to see all the endogenous protein in a single at a single cell resolution. We want to know where the signal transduction pathway actually occur at the different cellular subcellular compartment here. And there are several ways now to think about how to deliver that. And this is sort of one method that we try and it turned out to work pretty well. So we can use a CRISPR tool, for example, when to look at the endogenous insulin receptor level subcellular localization. We can use CRISPR to knock in there's a V5 tag and with half of the GFP. So they will generate protein, fusion protein, and this doesn't affect the function because the animal is alive and well. And then we can use anti-V5 to look at the global patterns of the insulin receptor, as well as to add a cell of interest, in this case the EMA neuron, express the other half of the GFP, and then they will reconstitute the GFP fluorescence, and then tell you where the protein is located. Okay, and this method can be extended to other color, and it does require a little bit of modification of that, so we have to go through another round of mutagenesis at the surface of that. The long and short answer is the GFP work out very well, and CFP to some degree, and YP doesn't really work. So at this point, we do have a two color, can look at this. And using this method, we can see that the insulin receptor do cluster onto, for example, photoreceptor growth cone during development. And if you look at the V5, then you see that broadly expressed but concentrated. But concentrated in the neural pairs here. So when we express, looking at a single EM developing DMA neurons here, and then look at the endogenous insulin receptor, quite surprisingly, we find the insulin receptor concentrated in the dendrite. If you turn this picture around 90 degrees, you see that insulin receptor signal concentrated in basically the central part of the dendrites. And we can extend this method to other systems. For example, we label NK hearing, we label Golgi with GM130s, and applications could be quite extensive. So since we find the insulin receptor is actually localized on the dendrites, not really at the cell body to receive the signal, and we wonder what are the source of the insulin or insulin-like peptides here. So this was work done in Larry Persky's lab using a single-cell RNA-seq, and to find out that out of 8 different insulin-like peptides in Drosophila, There's only DIP2 expressing laminar neuron L3 and L5. You can see the L5 neuron as cell body here extend axon. They're actually in the proximity of the DMA neuron, and the growth cone is very close to it, and presumably can deliver the DIP2 into that. So we express GFP tag of DIP2 in the laminar neuron and show indeed it can be carried from cell body all the way out to the tip of the growth cone and capable of delivering to the tip of that. And most importantly, if you remove the function, use RNA knockdown the DIP2 in laminar neuron or specifically in L5, then you phenocopies the 12 phenotypes. So lastly, we already show you that the insulin pathway function promotes expansion of the DMA dendrite, and the activin secreted from the R7 photoreceptor that through the transcription factor SMAC2 to reduce the activity of, to inhibit or to suppress the expansion of the DMA dendritic arbors. We wonder how these two pathways might interact. So we can remove both pathways by either remove TOR and remove R7 or some combination of that. So the long and the short story is that they do have an active effect. That means that if you remove both pathways, they become look a little bit more like a wild type, that is in average. And genetics would say they function in a parallel pathway. But surprisingly is that when you actually, even you have an average size about the same similar to the wild type, you show a much larger variabilities in here. Okay, and we can carry out additional statistic tests to show that indeed there's a difference in their variance, and suggesting that the reason why you need to have this kind of yin and yang expansion and contraction regulation is to actually generate a robust size of the dendritic arbor. So I think I run out of my time, and sort of just take 1 minute to summarize it. So I think through this analysis, what really gives us a new view about how dendrite might develop. The dendrite really living in a complex environment in the brain during development. And there's through a critical communication between the presynaptic partner or presynaptic non-presynaptic partner to deliver either diffusible by acting very short range to control the size of the dendritic arbor. And the reason for the DMA neuron to have both the positive and negative regulator in that is to increase the robustness of the size control. Okay, the communication between the presynaptic and postsynaptic partners not only limited to diffuse elements, but also involved in through the direct cell-cell contact. So in this case, the NK hearing to determine where the region the dendrite elaborate. Okay, so I'm going to stop here and acknowledge people who actually did the work. So the work described here are primarily carried out by John Lang, previous postdoc John Lang Ruo, and the junior team here, and now with the help of current postdoc current staff scientist Ziyang Lins and Borges. Okay, so we helped with when I was NIH, with the computational people to help me with the registration mechanism, and with the EM reconstruction was in collaboration with the EM target. I'll just stop here and take any questions you might have. Thank you.